Hello all, fellow peers. I hope you are all enjoying your day so far. This is the KNPE 429 present/teach a research article.
I'm Sean.
And I'm Andrew, and let's see what we're getting into today.
Okay, so this research paper is entitled Vasodilation is obligatory for contraction-induced hyperaemia in canine skeletal muscle, and was written in 2004 by Hamann, Buckwalter, and Clifford.
Okay, now we'll go into the overview of our presentation for you guys today. We will begin with some brief background information on the study, as well as a brief explanation on the underlying physiological mechanisms. We will then discuss the methods, i.e., what the researchers actually did in their study, the measurements they took whilst performing the experiments, and we will then discuss the results they found, as well as their and our interpretations of them. Lastly, we will discuss the possible limitations to the experiment.
Okay, so we all know that flow is equal to our pressure gradient times the conductance, and these mechanisms that are listed here on the slide alter blood flow by either altering the pressure gradient for flow between the veins and arteries or by altering the conductance. So vasodilation occurs based on either the metabolic or the red blood cell oxygen sensor hypothesis. Contractions in the muscle will increase the demand for oxygen and subsequently cause these vasodilatory mechanisms to increase the conductance for blood flow by increasing the diameter of the arterioles. So this allows for the increased blood flow and increased oxygen delivery to the muscle. So this is believed to occur in combination with the muscle pump. So the muscle pump increases flow by increasing the pressure gradient between the arterioles and the venules. The arterioles bring oxygen-rich blood into the muscle, while the venules remove the metabolite-rich blood from the muscle. So during contraction, the muscle is under a period of impedance to blood flow, as you can see in the picture on the right, entitled contracted skeletal muscle. Following this impedance period, when the muscle contraction ceases, there is an enhancement period in which the blood is pumped through the veins at a higher rate. The departure of blood through the veins acts as a pump, sucking more oxygen-rich blood into the muscle due to the increased pressure gradient.
So as we mentioned previously, the muscle pump and vasodilation are thought to work together in increasing the blood flow to the muscle following an increase in the muscle's oxygen demand. The researchers in the study aim to isolate the effects of the muscle pump by preventing vasodilation from occurring. They hypothesized that the initial hyperaemic response, or the increase in blood flow, was due to vasodilation as a result of smooth muscle hyperpolarization.
Okay, so just the protocol for the study here. The experiments had 2 sets of 6 dogs, which provided a very controlled sample for the study. The dogs were anesthetized and they were assisted in breathing with a mechanical respirator. The dogs were placed on a stereotaxic frame, which more or less allows them to be in a position that would mimic exercises such as running. So their legs were held in place to ensure that there was always isometric contractions occurring. The protocol was actually quite invasive. The sciatic nerve was cut and it was stimulated for a period of 2 milliseconds to induce brief isometric contractions. This contraction was performed under 3 different conditions. The first condition was a control contraction in which the nerve would be stimulated without any infusions of any kind. The second contraction had a potassium infusion, which was at the femoral artery, and the potassium injection was performed to prevent changes in the membrane potential of the smooth muscle, which would subsequently prevent vasodilation from occurring following the contraction. So the third contraction was an infusion of an alpha-1 adrenergic agonist, this was basically just done because the potassium infusion also caused vasoconstriction and a drop in blood flow, so they wanted to control for that while basically still allowing the membrane potential to be free to cause vasodilation via the conductive vasodilatory mechanisms. So they're looking at the change in hind limb blood flow. That was their main outcome measure following the contraction, and this was measured with an ultrasound at the iliac artery. So after the experiment had concluded, all the dogs were humanely killed with an overdose injection of potassium chloride.
So as you can see, these are the main measurements the research group took into consideration. Blood pressure was monitored to ensure that the dogs were still in stable condition despite being heavily sedated. Arterial blood flow was the main measure examined in the article, because of the reasons we discussed earlier. And lastly, muscle force production was measured to ensure that the infusion of potassium did not cause.
Okay, so without wasting too much time going into the full explanation of the mechanism here, but the conducted vasodilation occurs as a result of these pharmaco and electromechanical responses. So typically the change in membrane potential will cause calcium channels in the smooth muscle to close, which actually prevents smooth muscle from constricting. So that is what causes the vasodilation following a contraction. The process is initiated by just metabolites binding to a receptor, but we're not going to get into the full mechanism. But basically the infusion of the potassium will be aimed at causing a maintenance of membrane potential, which actually prevents these calcium channels from closing. So that will allow vasodilation to be sort of dampened.
Okay, so this is an original tracing of one of the dogs going through the protocol in which the blood flow is being measured directly. So as you can see, with the infusion of both potassium and phenylephrine, the baseline blood flow was lower than the control, or before the dogs received the infusion. What we can see that the researchers' hypothesis looks like it may be confirmed. With the contraction at the arrow, we do not see a very big increase in blood flow as compared to the phenylephrine condition, where even though the blood flow is attenuated before the contraction, we still see a pretty significant spike in the blood flow. So taking the time scale into consideration, which is in the bottom right corner of the figure, we must note that the response to blood flow in the potassium condition is attenuated, and we do not see that in both the control and recovery, as we can see by the large spike in blood flow.
Okay, so this figure represents the pooled data, or the combined results of all the dogs tested, and it's just a summary of the changes in blood flow. So we have 3 different phases in each of the 2 infusion protocols, one being control, one being the actual infusion contraction, and one being recovery. So the asterisk indicates that there's a significant difference between the potassium infusion contraction and all other contractions that were completed by the dog in terms of the change in blood flow post-contraction. So this was statistically calculated via a 2-way ANOVA, and this difference basically confirms our researchers' hypothesis that the rapid increase in active skeletal muscle blood flow with the onset of exercise is attributable to vasodilation as a consequence of smooth muscle hyperpolarization.
So initially there was a bit of debate to whether vasodilation accounts for the immediate change in blood flow, but this study provides some evidence to confirm that vasodilation does take a large part in blood flow increase following the increase in exercise. And not only does it take a large part in the blood flow increase, it also occurs quite quickly. So contrary to previous studies that had shown that vasodilation may only occur after 6 or 7 seconds, this experiment showed that the vasodilatory responses actually occur far quicker than that. Lastly, the muscle pump was thought to be insignificant in the immediate changes in blood flow following a contraction. This was proved by seeing no substantial changes in the potassium-infused condition's blood flow levels.
Okay, so just briefly to finish up, we had a couple limitations that we wanted to discuss to this study. Like, obviously, they weren't able to directly measure vasodilation, even though it's the primary focal point of the study. They didn't measure arterial diameter. They just measured the blood flow. So it may not be entirely feasible to measure the diameter of each capillary or anything like that, but we're still unable to determine anything more than changes in blood flow. We don't know whether the 2 mechanisms were completely isolated from one another during the potassium infusion. So the potassium infusion may have caused some sort of venoconstriction, which could have confounded the results. So also the second limitation, they used dogs, not humans, in their study. So obviously this was very internally valid study, but whether this is completely attributable to humans as well, we don't know. Whether our muscle pump may work in a slightly different way or something like that. So these results were very internally valid, but unfortunately some external validity was sacrificed just based on the study sample.
Thank you very much, and enjoy the rest of your day.